Let's talk about carbohydrate digestion. Not sure what this nutritional label is from, but there's a ton of added sugar in it. So I might avoid this product, whatever it is. Here we're looking at a molecule of glucose, which is our main form of energy or fuel that we use to make ATP in the body. Glucose plus oxygen gives us ATP. This is characterized by the formula C6H12O6. It does have 6 carbons, 1, 2, 3, 4, 5, and 6 carbons. And we certainly could count all the 12 hydrogens and 6 oxygen if we wanted to. But once again, that is a monosaccharide known as glucose. A monosaccharide is a single monomer or subunit. There are 3 types of monosaccharides that we consume in our diet: glucose, fructose, and galactose. All of them are characterized by the formula C6H12O6. Monosaccharides are the only form of carbohydrate that can be absorbed across the intestinal lining or the cells of the intestine. So we'll take a look at that as we move forward. We also consume disaccharides, which are composed of 2 monosaccharides linked together: sucrose, lactose, and maltose. Sucrose is a glucose molecule and a fructose. Lactose is a galactose molecule linked to a glucose. And 2 glucose molecules linked to each other is the disaccharide known as maltose. Disaccharides need to be broken down enzymatically before they can be absorbed. That is to say, they are broken down enzymatically into their monosaccharides, glucose, fructose, or galactose. And the enzymes that break down disaccharides are known as disaccaridases. The disaccharidase for sucrose is sucrase. Disaccharidase for lactose is lactase, and for maltose, it is maltase. So those are the enzymes that break down these particular disaccharides. The largest of the carbohydrates are polysaccharides, starch, cellulose, and glycogen. That is to say, the ones that we consume in our diet. Starch is a storage form of glucose in plants. Glycogen is the storage form of glucose in animals. Cellulose makes up the cell walls of plants. We consume it, but we cannot digest it nor absorb the constituent parts of it. So cellulose leaves our body in the form of fiber. Starch and glycogen, like disaccharides, need to be broken down enzymatically before we could gain access to the monosaccharides. That is initiated in the oral cavity with the salivary amylase, which starts breaking these polysaccharides down. Once we get to the stomach, the low pH of the stomach destroys and denatures that salivary amylase, so it no longer functions. Fortunately, when these polysaccharides enter the duodenum, there's another amylase from the pancreas secreted into the duodenum, the pancreatic amylase, that continues the breakdown of these polysaccharides. Once they're broken down into their monosaccharides, they can be absorbed. If they're broken down into disaccharides, then those aforementioned disaccaridases will break down sucrose, lactose, or maltose. When we get to the enterocytes, an enterocyte is merely the cells lining the intestinal wall. Over on the left here is the lumen. To the right is the extracellular fluid with the blood plasma. And when these monosaccharides are being absorbed, it's similar to what we saw in the proximal convoluted tubule of the nephron. That is to say, these monosaccharides are first going to cross the apical surface of the enterocyte and then the basal surface, getting into the blood plasma. Both glucose and galactose utilize the sodium glucose co-transporter to move into the cell. I should say utilizing the kinetic energy of sodium moving down its gradient for glucose and galactose to move into the cell against their gradients. Once they're in the cell, they can easily diffuse out via facilitated diffusion through the GLUT2 channels. To be clear, this sodium that comes in, creating the kinetic energy, will immediately be pumped out by the sodium-potassium pump. Now, one might ask, why is there a high concentration of glucose inside these cells when it's normally low in body cells? And that is because it is not converted to a different substrate like glucose 6-phosphate. And also, because these enterocytes utilize glutamine, which is an amino acid, to make ATP rather than glucose. Fructose moves into the enterocyte via facilitated diffusion, moving via the GLUT5 channel on the apical surface and then the GLUT2 channel on the basal surface. And once again, the molecule glutamine, which we see here, is the main source of energy to make ATP within the enterocyte.